In the previous video, we learned that the biochemical basis of life is similar in all living things, as the cells of living organisms contain carbon-based compounds that interact in similar ways. We learned that carbohydrates are an example of these carbon-based compounds. Other examples include lipids, proteins, nucleic acids, ATP, and water. However, in this video, we'll focus on lipids. A lipid is a type of organic molecule that is primarily hydrophobic and consists of carbon, hydrogen, and oxygen atoms. Lipids have many uses in living organisms. For example, they are used in the bilayer of plasma cell membranes, they are used to make certain hormones, and they are used as respiratory substrates. A respiratory substrate is a molecule that can be oxidized in a biological system to release energy for respiration to produce ATP. Lipids are also found around delicate organs to protect them, such as the heart. Some lipids are stored beneath the skin to help keep the body warm. And finally, lipids are used to make the myelin sheet in nerve cells. The myelin sheet allows a nerve impulse to travel more quickly along the neuron. You'll learn more about this in Year 13. There are two types of lipids you need to know about: triglycerides and phospholipids. Triglycerides are formed through a condensation reaction between one molecule of glycerol and 3 fatty acids. This leads to the formation of 3 ester bonds, producing 3 water molecules. Triglycerides can also be broken down in a hydrolysis reaction, which uses 3 water molecules to break the 3 ester bonds. This breaks down the triglyceride into a glycerol molecule and 3 fatty acids. Fatty acids are composed of a non-polar hydrocarbon chain with a polar carboxyl group at the end. A carboxyl group consists of a carbon atom double bonded to an oxygen atom and single bonded to a hydroxyl group. And a hydrocarbon is a chain consisting of only hydrogen and carbon atoms. At A level, the fatty acid chain in triglycerides is referred to as the R group. The R group stands for the residual group. The R group in triglycerides can either be saturated or unsaturated. A saturated fatty acid is a hydrocarbon chain containing only single bonds between carbons. An unsaturated fatty acid, on the other hand, consists of a hydrocarbon with at least one double bond between carbons. Each time we add a double bond to the hydrocarbon chain, it creates a bend in the chain. Also, the more saturated a hydrocarbon is, the more hydrogens it contains. Therefore, the more unsaturated a hydrocarbon is, the less hydrogens it contains. Triglycerides can take the form of either fats or oils, depending on the type of fatty acid they contain, saturated or unsaturated. Fats consist of saturated fatty acids, while oils are composed of unsaturated fatty acids. The structure of unsaturated fatty acids, with bends in their chains, prevents them from being tightly packed. On the other hand, saturated fatty acids form straight parallel chains, allowing them to be tightly packed together. Firstly, triglycerides have a high hydrogen and carbons to oxygens ratio due to their fatty acids. This is important for 3 reasons. First, it makes triglycerides a good source of energy. Secondly, it makes them a good source of water for respiration. Thirdly, it gives triglycerides a low mass-to-energy ratio. This means that they can store and release lots of energy in a small amount of space, which makes them great storage molecules. Secondly, triglycerides are large molecules and as a result, they can't cross cell membranes. Triglycerides are also non-polar, which means they are insoluble in water, therefore, they do not affect the water potential of a cell. In phospholipids, one of the fatty acids of a triglyceride is substituted by a phosphate-containing group. Given that, a phospholipid consists of a glycerol molecule attached to 2 fatty acids and to 1 phosphate group. The fatty acids bond to the glycerol molecule via 2 condensation reactions, forming 2 ester bonds. Phospholipids have 2 distinct regions. The phosphate head, which is polar, and the tails of the phospholipid, which are non-polar. The glycerol molecule plus the phosphate makes up the phosphate head and the 2 fatty acids make up the tails of the phospholipid. Before we dive into the properties of phospholipids, it's important to understand a key rule about polarities. When it comes to polarities, like, dissolves like. This means that polar molecules are attracted to and will dissolve in other polar molecules, while nonpolar molecules are attracted to and will dissolve in other nonpolar molecules. We've seen that the phosphate head in phospholipids is polar, meaning it can dissolve and mix with water molecules, since they are also polar. As a result, these heads are considered as hydrophilic. On the other hand, the phospholipid tails are nonpolar and do not mix with water. Instead, they repel water, making them hydrophobic. Because phospholipids have both a polar and a nonpolar region, when they are dissolved in water, they orient themselves in such a way that the heads are directed towards the water molecules while the tails face away from them. This orientation can lead to the formation of one of three structures. First, they may form a bilayer, a structure consisting of two layers of phospholipids that make up the basic structure of cell membranes. The second possible structure is a micelle, which is a spherical arrangement involved in the absorption of fats. Lastly, they might form a monolayer, a single layer that forms on the surface of water molecules. In a phospholipid bilayer, the hydrophilic heads face outwards towards the water molecules on both sides of the membrane, inside and outside of the cell or organelle, while the hydrophobic tails face inwards, away from the water, and interact with each other. A bilayer allows phospholipids to do two things. Firstly, this arrangement creates a semi-permeable membrane that acts as a barrier, allowing certain substances to pass through, while keeping others out. This is because plasma membranes contain phospholipids with a combination of unsaturated and saturated fatty acids. Unsaturated fatty acids, as we've seen before, can't be tightly packed together. As a result, they form gaps in the bilayer, which makes the membrane more flexible and allows the selective movement of certain substances. Secondly, the bilayer also provides a fluid environment for lipids and membrane proteins, which can serve various functions, such as transport, signaling, structural support, and cell recognition. Glycolipids are a type of lipid molecule involved in cell recognition. They consist of a phospholipid with one or more carbohydrate chains attached. Glycolipids help the body distinguish between self cells and non-self cells. Additionally, they serve as recognition sites for other cells, allowing them to bind to each other and form tissues. Both triglycerides and phospholipids share one more property. Despite having a polar region, phospholipids are generally considered nonpolar because their nonpolar region is larger than their polar region. Therefore, both phospholipids and triglycerides can dissolve in alcohol, which contains both nonpolar and polar regions, allowing it to mix with both types of substances. We use the fact that lipids dissolve in alcohols to test for them. This is known as the emulsion test. Here's the procedure. Add 2 cubic centimeters of the food sample to clean, dry, and grease-free test tubes. Add 5 cubic centimeters of alcohol to the food sample and shake the test tube thoroughly. Then, add the same volume of water and gently shake the mixture. If the sample contains lipids, a milky white emulsion will form. An emulsion test is an example of a qualitative test. This is because it tells you if the lipid is present or not in the sample.